Buenos días. Disculpar este retraso, estas incidencias técnicas. Son los problemas del directo, ¿verdad? Bueno, excepto los que lo vean después en diferido. Es realmente un honor. It is a great pleasure and an honor to have here today Professor Tatsuo Kaneko and also Dr. Maiko Okajima, both of them belonging to the Japan Institute for Science and Technology, which is one of the biggest organizations for science in Japan. And yes, I will. Yes. So, Professor Kaneko is Dean of Energy and Environmental Area and at the Graduate School of Advanced Science and Technology, and is the head of Excellence Core Research Center of Sustainable Materials. This institute is located in a very pleasant place in Ishikawa, which is not far from Kanazawa, in the west of Japan, and it's a very nice place and a very important institute. Professor Kaneko obtained his PhD in polymer chemistry at the Tokyo University, and later on was working at the universities of Hokkaido, Kagoshima, and Osaka. And after that, he joined the School of Material Sciences at the JACE, the Japan Advanced Institute of Science and Technology, and then was promoted to full professor. And he also joined the bioengineering department of the University of California in Los Angeles as a visiting associate professor. And the current interests in research include soft matter, gels, polyelectrolytes, biodegradable plastics. And he has published more than 200 papers in high-impact journals, including Nature, Materials, and so on. He has received various international awards from the ACS, the UPAC, from Germany, companies, among others. And he's currently in charge of the project, a big project, which is financed by the Japanese government and is devoted to biodegradable plastics, which is in direct relation with the big problem, of pollution by microplastics and so on. And today, Professor Kaneko will introduce the talk entitled like Moonshot, Seeing in Bioplastics with Environmentally Switched Marine Degradability, and the co-author is Dr. Maiko Okajima, here present also. Thank you very much.
Okay. So, okay. Thank you very much for very kind introduction. So I'm very happy to be here with you. And then so I think that 10 years before I went here. And then so I remember it was very nice time. And this time, I'm looking forward to seeing both of you, and then some other colleagues and your students. And then so I also very happy now making a lecture about bioplastics. So especially that is degradable in marine environment, but that is very useful during use. Okay, if degradable during use, that is cannot be useful. So that means a switch of the biodegradation. That is very important. So I want to introduce now these researches. Maybe. Okay, that's good. And before with today's topic, I want explain another topic for my research background. So actually, my research background is very, very high performance polymers. And then, so together, I'm now trying to preparing the very high performance and others degradable polymer. Actually, that seems opposite, but that is a single line. One is one edge, another is another edge. Okay. This is a very high performance polymers materials because this aromatic, and then aromatic is very hard molecules, and then that's having the amine hydroxyl COH. This have already synthesized by using the bio or that's microorganisms. Okay. This is called the streptomyces species. That is exist in the soil, inside the soil. In Japan, in Spain, both. Any places this streptomyces is exist, and then so producing this kind of the biomolecules. And then if this is converted by chemical technique, this OH become the NH2. This is a very special chemistry, that's the SMILES rearrangement. And then so this successfully get these materials, and then this can be converted to the polybenzoimidazoles. This structure is this aromatic. Here is also aromatic. So heterocycle, benzo-heterocyclic structure. So that is all aromatic, conjugated materials, and then this shows the ideal structure. And then so very high heat resistance temperature around 700. Okay, this 100% means homopolymer of this one. And then this shows very, very high, this 700. And then on the other hand, this one, this is aramid. Aramid is very famous for bulletproof. Okay, for that's a policeman use. Okay, and structure of this one, this aramid here, and this is 500 degrees Celsius. This means 10% mass loss temperature. Okay, the if polybenzoimidazole and the aramid is mixed, and then this copolymer is prepared. Okay, and this copolymer shows the various mass loss temperature. My imagination at the beginning, from here to here, this simple line. Okay, now this is a very easy consideration, but a little bit, okay, but very small amount of aramid is incorporated into the polybenzoimidazole. Okay, around like this. Okay, only the 15% is incorporated. So mass loss temperature increased and then decreased. So as a result, the higher mass loss temperature than homopolymer is produced. So this is actually the highest temperature on record. And then so this molecular design is now a very nice design. And then that is derived, this is processed into the film. Okay, and if you have some interest for this one, I can send you as a sample. Okay, and then so for microplastic. Okay, so do you know who? This is Makoto. Yeah, maybe several months before he also made a lecture here, I think. And then so actually very good friend. And then this is Professor Takahara. And then so as a Thai friend, and they searching something. This is microplastics. Okay, in Thai. This is September, and then we found some microplastics. Actually, every place in the world you can find microplastics. Or some of them is just macroplastics. Yeah, this is 10 centimeter. This is macroplastics. That's also so much. These kind of the net, that's too bad. Yeah, that's induce the ghost fishing. That's too bad. And then these kind of macroplastic can be converted to microplastics. in time. Okay, I think that maybe several years later, that's become microplastics. Okay, and then so macroplastics and microplastics, both in total, the 8 million ton per year is added in ocean annually, as you know. And then so various kind of the ghost of fishing, that's very bad issues that is made. And then so if that is become the microplastics, and then that's swallowed mistakenly, and then so they will die, and the ecological issues have happened. We have to solve this problem. Yeah. So several years before, I think the 19th century, and then this kind of the aliphatic polyesters have already developed by some companies such as this Kaneko. I think that's now become very famous. But my name is Kaneko. That's a little bit similar, but completely different. Okay, this is some kind of the girl's name, but different. Okay, this means chemistry. This car is a chemistry. Okay, but then that's the Kaneko that's developed the PHBH. This is also a kind of the aliphatic polyesters, and then degradability very nice. This also degradability a little bit slow, but very nice. And then so this is degradable in soil also. Okay. But in soil, degradability is a little bit faster comparing to the marine. Okay. Marine have the very small amount of the microorganism, and then so speed is very low, so very difficult to develop the marine degradable polymers. Okay. But we can produce the degradability in marine environment. And for example, this one, here, this one, this is a structure of the protein. So this is composed, the living body. For example, here, this is a protein. Okay. And then structurally, you can find a structure here, around here, this amide group, and then carbon number 1, 2. So this is a nylon 2. So you are composed of the nylon, okay, but the degradable nylon. Okay, and then so if the carbon number increase, for example, this 1, 2, 3, 4, nylon that's become nylon 4. Okay, nylon 4 is actually the degradability is very nice, and the producibility is also very nice, and then that is also shows the degradability in marine environment. Okay, that is very good, but performance not so good. So that cannot be used in such like this. Cannot use. Okay, so and then nylon 66. That's very useful. Maybe you know, this is some of the cloth. That's fiber is derived from nylon 6 or nylon 66, and then that is already distributed in the society from the ancient days. And then, but this is not degradable. Okay, that's degradability and then usefulness. That is balance is very important. And then recently, so Professor Kato and then Professor Negoro, that's Japanese, that's found the enzyme for can be used as a catalyst for degrading nylon 6. Yeah? So that discovery is very good. But nylon should be the oligomers, so molecular weight should be low in this case. If the molecular weight become high, the degradability become very slow. So another enzyme should be established. But recently, he is my very nice collaborator. Recently, he are now developing the very nice enzyme. Okay. I want to say that this is degradable but not useful. This is useful but non-degradable. And then so around carbon number around 4 to 6, okay, around here, that is very important for preparing a switch from useful to degradability. Okay. And then I found from this list. This list is okay. This is listed by the USA Department of Energy. And then, so these molecules have already fermented in the mass level in the society, in the world. And then, but all of them shows the not so high, actually the nice structure for me. So, but this one, itaconic acid, that's have the C carbon number 5, the 1, 2, 3, 4, 5, and then so that's double bond is very rare case. Others have not carbon-carbon double bond. Okay, only itaconic acid have the carbon-carbon double bond, and then so many people have an interest for this structure, and then they preparing the polymer by radical polymerization or some other kind of the living polymerization. But my interest is a nylon, so we use this carboxylic acid, and then this carboxylic acid, and then that can be polymerized by some polycondensation method. And then, so for example, polyesters can be produced by other researchers, but from us, nylon is produced from this. And then the itaconic acid have already fermented, very, very cheap, okay? This is a little bit expensive comparing the petroleum process, but the other biomolecules much higher price, okay? And then this is very good. This is structure of polyesters. So our consideration, okay, this nylon can be produced very easily, but this is not so good, okay? So this side reaction occur. This nitrogen have a very high reactivity. So I mean the nucleophilicity, that is very high, and then this shows the addition reaction to this double bond, and then so this side reaction occur. Okay, in this case, so around these molecules, more than 3 diamine exists around here. In this case, this side reaction occur. So we have to mix in the molecular level homogeneously. So we tried to produce, okay, this one, salt monomer method, okay, and then salt monomer method is very famous in the nylon synthesis, and then this is the itaconic acid, this is the acid, okay, and this is a base. If this and this mix, this ion pair is produced, and the ion pair is now continuously now that's connecting, okay. And then so this ion complex can be get as a white crystals from this ethanol mixture. And then we can this filtered and then dried and then heated around 200 degrees Celsius without any organic solvent. And then so this can be converted via micro addition and then cyclization, and then this pyridine ring can be formed. And this pyridine monomer have acid and then amine, and then heating more. In this case, this nylon can be produced. This nylon structure is very new because this cyclic, okay, heterocyclic structure is incorporated in the main chain. So some people know PVP, polyvinyl pyridine. Polyvinyl pyridine is a side chain. This is a main chain. So that shows the very good properties for metal binding or something as absorption properties. But this main chain, and so that shows the very high melting temperature or mechanical properties. Okay, and then we have already got this nylon fibers. Okay. This is explanation of the reaction mechanism. So that's a little bit, it takes a long time, so I will skip this slide. But if you have an interest later, please ask me. Okay, and the resulting these properties, thermal properties and mechanical properties summarized in this table. And then this is the carbon number of the diamine. Okay, itaconic acid carbon number is C5. But diamine can be varied. Okay, this is ethylene diamine. This is hexamethylene diamine. So this can be compared directly to the nylon 6. Okay, nylon 6 shows a glass transition temperature around 56 degrees Celsius, but our nylon that is 97. So this is much higher melting softening temperature. This means strength at the break under the mechanical test. Okay? This is also very good. This is almost the same with other conventional nylons, nylon 6 or nylon 6. So this is very useful. Okay? But this is very useful but degradable. Okay? And then this is a test in soil. Okay? For example, this white matters, that is polylactic acid. That is very famous biodegradable polymer, but that shows a slow degradation. For example, if we use this around here, that's soil. The soil is used. This polylactic acid shows the 16 weight% decrease. That is degradability. But our bio-nylon, 98% disappear. Okay, this is completely disappear. This is a diamine carbon number. Okay, even if we use the aromatic one, that's diamino diphenyl ether. Okay, if that is used, the 34 weight% is degrading. Okay, and then so this very fast degradation in the soil. So because this shows the this conversion. Okay. And then this is a pyridon ring. This pyridon ring shows the ring opening reaction and then the water in the presence of the enzyme, soil enzyme or some microorganism in soil. Okay, and then this is a hydrophobic structure, but this is hydrophilic structure. So hydrophobic to hydrophilic conversion occurred inside the soil, and then so some of them is disappear. That's dissolving in rain water, okay, during the 1 year, and then that's it disappear. Okay, but we cannot get the disappearing matters from the soil. That is difficult, and then so we use the laboratory technique. Okay, this is petri dish, and inside here the water. We cannot see very well, but this is water. Okay, and then in the central one is nylon. This is nylon and C5 plus itaconic acid. Okay, and this is if this is irradiated by mercury lamp, the UV irradiation, and then this is the intensity is very high. In this case, from here, this is size is 5 millimeter, a very small one. If this is disappear after 6 hours irradiation. Okay, this is very strong, so that means if you use sunlight, this 1 hour means 1 month, okay, in the environment case. So 6 hours means 6 months, that will disappear, okay, by UV light irradiation. Okay, UV light also, that means sunlight also very small amount of the UV light, UV range wavelengths including. So this shows the ring opening reaction inside the water. So especially seawater is very nice action. So seawater, because seawater is have a small amount of alkaline. Okay, that's alkaline catalyze this ring opening reaction like inside soil. Okay, so marine environment also very nice from this conversion from here to here. Okay, so during use you can use this structure. This is very stable, and then but after going to the marine environment, the sunlight continuously. Okay, and then seawater, that's very nice environment for ring opening reaction, and then so this chemical conversion occur very easily. The inside the laboratory, this non-methylene diamine or meta-xylene diamine, the structure different, and then so time is different. This is very long. Now I summarize this time by volume. This is volume of the resulting volume to the initial volume. Okay, then that is increased because water swelling, and then suddenly disappear. That's plot disappear. In this case, you can compare the aliphatic carbon number length. Non-methylene means C9, very, very hydrophobic. Yeah? So very, very slow. But hexamethylene diamine, that's very fast. Yeah? But even if the very slow non-methylene diamine, terephthalic acid is incorporated, I think that is more strong, and then that is shows you more slower, I think. But the result is different. If terephthalic acid is incorporated into the non-methylene diamine, that's from here to here, that's accelerated. Okay? Because we use the UV light, and then so terephthalic acid very easy to absorb UV light, and then so that's excited, and then that's photo energy is very easy to convert to the carbonyl group and ring opening reactions. For example, this here, this have the pi electron around here, and then sunlight or something as a UV light that is excite here, and then so this is exciting state, and then this is a resonance between the charge separation and then biological state, both state. That's both state is reactive. In this case, the OH minus can easy to react in the marine environment. Even in this case, that's water can react, and then so resulting ring opening reaction occur under the sunlight. In the case of the alkaline solution, this is capped. COH can capped, and then this cannot back to the ring closing. So we already tried in the acidic solution. If that is dried inside acidic solution, this is H, and then that's become back to the ring closing reaction. Okay, so and then so in this case that is very useful. One reason for useful, we use in the dry state, and then not the alkaline state actually. In this case, if we use the washing by that's mean laundry, and then so in that case, this even if that's ring opening reaction, after dry that's become ring closing, and then so this unstable state do not kept. Okay. So if the degree of this ring opening reaction occur, so this hydrophilic matters that is accumulated continuously long time, and then so as a result of the 6 months, that's become disappear inside seawater. Okay, and then so from here I can explain the degradability, biodegradability. Okay, this structure is not biodegradability because if we use the seawater, this SW means the seawater. This is sediment. That means mud at the bottom of the sea. And then that's activated. Seawater is activated, and then we use the itaconic acid-derived nylon. The carbon number 6, I means itaconic acid. Okay? If that is put, and then this structure, but the background is blue, and then we use the nylon, become orange, but the same. This is non-degradable. So this is BOD. So microorganism that demand the oxygen for degradation. So this is not to degrade. But on the other hand, this structure, if we use the nylon 6I, 0.1 gram is here green, and then this is 0.5 gram. Okay, and then so this biologically demand oxygen that is increased. Okay, this is background. So this is the evidence of the biodegradation of this structure. So we appear this is a degradation photo switch, non-degradable, degradable under the sunlight. Okay, and then so this is converted into the degradation degree in the y-axis. Then if you use the 4 days, and then that is become the 70% degradation shows. And then so what kind of bacteria degrade these itaconic acid derived nylons? Yeah. This is the answer. So, gamma proteobacteria and the alpha proteobacteria works very well to degrade our nylons. That is found in the sea water. Okay. And then so we have to check the safety, okay? And then because our nylon become gradually hydrophilic, that is dissolved in the seawater, and then if that's toxic, very easy to, yeah, that's bad, very easy to be bad for the marine ecological environment. And then so we use this method. This is always a plankton. So that's this structure is very nice for degradable nylons. Okay, and then so that can be incorporated into the existing nylons. For example, nylon 11. Nylon 11 is derived from this 11 amino indicanic acid. That is have already circulated, and then so this is incorporated by itaconic acid and then hexamethylene diamine. And this copolymer can be produced. So in this case, various kind of the materials that can be successfully produced. So producibility is very good. And for example, the 3D printers we also tried, but this is a robot arm printing. Okay, here is our nylons. Okay, here is materials for 3D printing, and then so around here 200 degrees Celsius, and then that is cycling, and then this fibers now coming here, and then this can be processed. Okay, so this can be used for the 3D printers. Yeah, producibility very nice. And then so resulting toughness is very good. Toughness. This value is almost similar to the nylon 11. Okay, nylon 11 toughness is very famous. That's very good. That can be already used inside the engine, around the engine. That's an oil pump, kind of that's a tube for oils inside the vehicles. And then but the nylon 11 shows no degradability. So if we use the itaconic acid, and then that's become the disintegration from here to here, and then that's become the biodegradable. Okay. So and then so we also check the artificial stomach model. That this water, including the pepsin, and then HCl pH2. Yeah? And then so this matters is our nylons. This disappear after 16 days, and then so this film also disappear, almost disappear, but become disintegrated. And then the molecular weight is also changed from here to here. That's become small, small, small. So that's also digesting matters of the nylons. So that is edible nylons, but I didn't try. And then so sorry, that's the same one. Okay, I also appeared in the TV, but now this is not included. And then so this additive, so maybe you're familiar, this monomerite. That mixture of our bio-nylon is very nice. We use the sodium MMT or covered by the something like a OH type surfactant. Okay, that's also aromatic type surfactant. And then so after mix, and then we can process, and then check the. Okay. Sorry. Oh, cannot work somehow. Oh, sorry. So maybe. Here, click, and then this one. Oh no, sorry. Okay, good. Thank you. And then so this resulting. So the mechanical property become very nicely enhanced. Okay, so this is toughness. Toughness is calculated by the stress strain curve area. Okay, and that is enhanced by using the sodium MMT. Okay. And then so because this is our consideration, so this is our first structure, and then so sodium MMT is alkaline, and then that's ring opening reactions, and then so many sites of the hydrogen bonding appears. Okay. And then so we want to produce these kind of the materials or safe materials for baby, or safe materials for sea animals, okay, and digestible in this case become the very nice, okay. This is just, and then so I'm now showing this one, this, yeah, and then so yeah, maybe it's a very rare shrimps around them. Maybe in Spain, so also very nice. And then so already professor have already tried. For example, this is crabs. Yes. Okay, this is a picture, very old picture. Yeah, this is Makoto. Yeah. And then so that's so many. This before eating, and then after eat like this. And then so and the 3 even the crab. So this, but Makoto 4. Yeah. Okay. So now this is topic of the biodegradable polymers. So I can introduce the this Sakura study. But sorry, so that is written in Japanese. So and then so I can use that as picture or data. That's okay. Thank you very much. So this is a name of the Dr. Okajima. Okay. And then so I can explain the by using this one. So this is a resource of the saccharine polysaccharide. Yeah. And then so that is have already collaborated with this group. Okay. And then this name is Suizaninori, but that is aphanotic saccharine. That is a scientific name. And then if that is observed by microscope, so we can find many cell, okay, unimolecular, unicellular, okay, microorganism, and then between the cells, we can find some space, including here the saccharine have even exist, and then so saccharine have can be extracted by the alkaline method, and we can find this one as like a cotton materials. And then so this is fibrous materials. That shows the molecular weight very, very high, 2.10 to the 7th power. So that's very high. And then so that is shows the very high viscosity, even if the this shows the 1%, but the 0.5%, 0.1%, that also shows the very high viscosity. And then so that shows the water retention capacity was also very high. And then so this resource of aphanotic saccharine have already cultivated inside some field of the Kyushu area of Japan. And then so inside of the laboratory, that cultivation have already succeeded. And then if that is dissolved in water, that becomes viscous solution, and then that is a little bit turbid, because that is shows the liquid crystalline, okay, and then that means that's the orientation domain already that is formed, and then this line is boundary of the orientation. Okay, this is evidence of the liquid crystallization, and then from here. If this is casted to get the film, okay, this film already this time also this is oriented. This is in plane orientation, and then if this is very thin, that is become flexible, and then so that can be easy to put the living body, and then if that is put inside water, and then that's become more flexible. And then so that's become gels, okay? And then before making gels, if this is heated, annealed, okay? And then so that's gel water content, softness have already controlled, okay? And then so we have a picture, but that means that this is gels, and then this gradually swollen in the movie. Okay, sorry, the inside here, that can be easy. I think that's not including, so I can try to check. Maybe cannot. Sorry. Ah, sorry, maybe. Sorry. So maybe again. So. Here. Here. Here maybe. Here. Okay. Good. Thank you. Yes, and then that gels shows the some. Okay, sorry. Okay, sorry. So back is here.
Probably, I don't know. No.
Yes. Okay. And if this gel is used for the DDS, that is gradually release the drugs. Okay, that's also did. And then so that is have checked the wood healing properties. Yes. And then so that's now trying to check for human body this is African medical doctor, and they tried. Okay, and then that's have the very nice results. And then so okay, I'll skip this one. Yeah, and the structure Sakuran is like this, but the Sakuran structure is a little bit complex. We are now trying to check by the X-ray scattering method by using the spring 8. This is a very big this synchrotron radiation source, and then very thin solution is used for Sakuran. Sakuran structure is controlled in the very, very low concentration, and then so this scattering strength is very low. So we have to use the very strong X-ray. Okay, and then we tried by using this one. Yeah, this one, and then the structure checked. Okay, and then so some diffraction occur, and then so we can make a fitting to get the structure of the Sakuran, and then so some fractal shows. Okay, like this. Okay, and then so the resulting is this one. The Sakuran length. Okay, that's contour length of the Sakuran is very high, and then that's more than 100 nanometer. That means a very big structure. Okay, like this. This is a Sakuran, and then so Kurisara have already found this kind of coiled structure of the Sakuran, so by using the SEM method. So I'm sorry. So now today, so that's missing. Yeah, coiled structure like this. That's very beautifully shown. Yeah, that's a little bit you can find in the FM, but in the SEM, that's also can be seen. Yeah. Okay, this is now introduction of the Sakuran. Okay, thank you very much. And then so today, so our topic is this nature-derived Sakuran, and then bio-derived nylons. Okay. And then so I summarize these two topics. Okay, thank you very much. Yeah.